Amanda here, and this week's rabbit hole had me once again looking at the infamous folate cycle and looking at it not through a new lens, but looking at it to answer questions I often have myself about when we use supplements. Do we actually understand the impact and the influence of the supplements that we're taking ourselves? Or if you're a clinician, how that supplement works in the body of the individual that you're treating. And I do, as a clinician, use supplements. I use them in a very, very targeted way. I very rarely will use a supplement that has a ton of ingredients in it, regardless of how good it may look, because I want to understand the impact of the different doses in the supplement on an individual's physiology, on their biochemistry. So because I work in genetics, because I validate what I'm seeing in genetics with lab testing, I'm literally able to see, like many clinicians, where the inefficiencies are in the biochemistry and how we can support bringing that biochemistry back online or up to par using food and very specific supplements, the ones that basically fit the need of the person I'm working with. So how do we know if we're an individual, if we're taking the right supplement, if we have a supplement that's formulated at a physiological level, one that's compatible with us and our genome? So this is my rabbit hole, and I want to take you into a couple of papers that really validated a lot of my thinking, but also gave me more things to think about. So I said we go into the folate cycle, so let's go take a look. So those of you who love biochemistry or recognize this, those of you who don't so much may be shuddering. But the bottom line is what we're looking at here is the folate cycle. And what does the folate cycle do? Basically, it has a couple of very, very important points or goals. One of them is to produce the 5-methylene tetrahydrofolate molecule, which is integral to producing methyl groups. It's passed on to homocysteine to regenerate methionine, which is then biotransformed to acetinosyl methionine. We call this the universal methyl downeth. So that's one of the major goals of the methylation cycle. The one that's missed, and I think it's just so important, one that I emphasize all the time when I'm working with and teaching and mentoring clinicians, is this reaction. And it's the reaction that involves this molecule, 5,10-methylene tetrahydrofolate, in the methylation cycle, in the folate cycle. So 5,10-methylene tetrahydrofolate has two destinations. It can be biotransformed by the infamous MTFHR to the 5-methyl form of folate, the one that we're all targeting, right? But see how here it also drives this really, really important reaction by which we are actually producing thymidine, which is one of the 4 base nucleotides for DNA synthesis. So 5,10-methylene tetrahydrofolate is as important for nucleotide synthesis as it is for producing the 5-methyl folate form of folate, that molecule. But this often gets missed out. It's like, oh, there is that part of the folate cycle. And as it turns out, it's extremely important because it's involved essentially in DNA synthesis. DNA is the recipe for life. So if this reaction is compromised in any way, can you see potentially how that could lead to DNA damage or something else which is more prevalent here, which would be the misincorporation of uracil, starting molecule, into DNA. In other words, if this reaction is not driven, we can have elevated levels of uracil, which, as it turns out, are pretty detrimental to DNA. To that point, what we're trying to do is drive this reaction. And as you saw, 5,10 methylene tetrahydrofolate is integral to driving this reaction. It's another very, very, very important reaction or kind of product, if you will, of an efficient folate cycle. But as the authors in the paper, deoxyuracil in DNA in Health and Disease point out, quote, genomic instability has long been implicated as a primary causal factor in cancer and diseases of aging. The genome, so the sum of all our genes, is constantly under attack from extrinsic and intrinsic damaging agents. Uracil misincorporation into DNA and its repair is an intrinsic factor resulting in genomic instabilities and DNA mutations. Additionally, the presence of uracil in DNA can modify gene expression by interfering with promoter binding and transcription inhibition or upregulation of apoptotic proteins. So I think this is a really wonderful graphic to show you, wow, look, here's the DNA. Look how these uracil molecules are incorporated into the helix. That immediately generates a repair mechanism, and you're left with potentially nicks in the DNA, which can be damaging or mutagenic. This potentially can be avoided if we drive this reaction, which requires 5,10 methylene tetrahydrofolate from the folate cycle. Moving on then really quickly, what got me really thinking, going down this rabbit hole, is a wonderful paper that was sent to me this week. It's an older paper from Michael Fenich in Australia, incredible researcher, where he was looking at or asking the question, do we really understand how to pair nutrients with or formulate supplements that would be consistent with an individual's DNA? This was in 2010. And he made the point here in this table directly from the paper I cited here, that these nutrients are not just folate, but folate is integral to the maintenance and methylation of DNA. So remember, methyl groups are driven, the production of them is enhanced, or the precursor to the universal methyl donor SAMe comes from the folate cycle. And so methyl groups are instrumental to methylating DNA. So that's one point. But as he makes the point here, that folate particularly is integral to the synthesis of thymidine from uracil and the efficient recycling of folate. He makes the point again, uracil misincorporation in DNA can result in increased chromosome breaks and DNA hypomethylation, which essentially won't have that methyl group to perform the role often of silencing DNA and DNA transcription. So then it also made one additional really important point. So we know that folate cycle is so important for driving thymidine synthesis. I've made that point a few times now. But he also makes this incredible point, which made me think about supplementation. And perhaps the slide there should say, when we over-supplement or we are inefficiently supplementing, can we cause genome instability? So he makes the point, it was actually brilliant, that if we over-supplement MTHFR with its cofactor riboflavin, which is vitamin B2, it catalyzes the irreversible conversion of 5,10 methylene tetrahydrofolate to 5-methyl tetrahydrofolate. What on earth is he talking about, and what does this mean? It basically, we'll go back one more time and look at the folate cycle. Basically what he's saying is if you drive the reaction forward to the 5-methyl tetrahydrofolate form of folate, that has a goal. But do you then lose availability of the precursor model, 5,10 methylene tetrahydrofolate, to drive thymidine synthesis? That's really what he's saying. And when that happens, as we saw in the previous paper, we can end up with a case of much higher levels of uracil, which when they get incorporated into DNA can cause significant damage. But just to finish off there and go back to this cycle, basically what it's saying is so often, as we know, we have MTHFR. Everyone has MTHFR. Some people have SNPs, some people don't. That's one gene that we look at in the folate cycle, just one. There's so many that are involved in this cycle and the handoff to the methylation of the methionine cycle here. But as he's saying, like if you push this gene, like, ooh, somebody's under-methylating, so we're going to give them a whole dose of B2 riboflavin to drive to really push MTHFR because that's its cofactor. Are we then driving a reaction here that's irreversible? Because this does not back up. You cannot retroconvert the 5-methylformine tetrahydrofolate to 5,10-methylene tetrahydrofolate. In other words, the horse left the stable, the train left the station. It can't go backwards. So that's my rabbit hole. Lots of food for thought. Again, I do supplement, but when I'm looking at biochemistry like this, it reinforces the need to understand what works with each unique individual and not to just supplement without monitoring, whether you're a clinician, whether you're an individual who's really invested in their health. So that's my rabbit hole.